All right, so we're going to move on to our next presenters, and they will again be tackling the question, or will be providing another perspective on the question of how you drive digital endpoint development, really with targeted at capturing meaningful aspects of daily life, combining aspects of qualitative interviews, sensor-based 6-minute walk tests, and at-home monitoring to give a full perspective of how conditions and treatments, or how conditions affect patients and treatments make a difference. They will be sharing their strategy that they have been developing for congenital myasthenic syndromes. I'd like to introduce Kim Offergeld, franchise clinical operations development lead at argenx, and Jan Clay, who is director of science at VivoSense.
Thank you very much for the invitation, and I'm very happy to be here. In fact, I'm following up on a first discussion last year where my colleague Stan already presented with Jan. Last year we presented on a different indication, and this year I'm actually very excited to take you through the data of our proof of biology in congenital myasthenic syndrome. And before I start with CMS as the indication, I first want to give a word on our molecule, adimanibart, or argenx 119, which is actually a very cool and innovative molecule with a very special and specific, unique mechanism of action. So within neurology, you have the motor neuron and you have the muscle, and the motor neuron talks to the muscle through means of your neuromuscular junction. And that's exactly where the 119 molecule is going to impact. Knowing that, we wanted to be very mindful and thoughtful of what is our first indication, and more in particular, the first experiment we want to actually test out to demonstrate whether or not our 119 molecule could mean something, not only for this indication, but could potentially also unlock other indications. So that's what is 119 about. Let me take you to CMS as the first indication. CMS is an ultra-rare neuromuscular indication. There are only about 300 people in the US suffering from this disease. It's a genetic disease, and there are no measures at all and no treatments. That puts argenx as such in a very unique situation to actually see what 119 could do. No measures, meaning that we actually had to pick measures from another indications, and we decided to take the measures from myasthenia gravis, the MG-ADL and the QMG. But we didn't think that that was enough, and based upon patient panel interviews, we actually learned that ambulation and walking would also be something of importance to measure. And that's how we ended up with also looking at the in-clinic measure, the 6-minute walk test, and also the actigraphy, so to look at the remote monitoring. We were able to actually not only demonstrate that the 119 molecule was safe and tolerable, but we were also successful in demonstrating a clinically meaningful increase in the 6-minute walk test distance, as well as the cadence. And so today's story is actually to take you through our evidence generation machine in different ways. And we're going to do that in a very structured way. We're going to share with you how we generated the data for the through qualitative interviews, not only to demonstrate that patients had a positive experience. So what we did was, at the end of a 12-week treatment, we had a structured interview with the 16 patients in this phase 1b trial. And through those interviews, we learned that they had a positive experience through the study. Not only that, they improved their symptoms. They also had an overall satisfaction of the clinical measures, and there was also an increase in mobility, again to demonstrate or to link back to our mechanism of action. And that increase in mobility you can see by some of the statements that are shared by the patients here, which is, I couldn't walk from the bathroom to the sofa. I had to stop. Now I can tell I have more energy in the legs and I don't stop. Or another one, which I personally found very powerful, was, I was able to go for a short walk, arm in arm with my husband, my daughter, which are things that are very dear to me. And it's actually those statements through those interviews that demonstrate that we did not only capture what was meaningful for the patients, but it actually was also that we're measuring the right things. And that is also what we did not only at the end of the study, we also did in the beginning meaningful aspects of health. And through those meaningful aspects of health, we measured or we asked the question, what are you expecting? What are your current symptoms and what are you expecting, so that we could tie those to the measures. Not only the digital measures, but we actually were able to complete the picture of how the DHT would be integrated amongst the family of other clinical and performance-based patient-reported outcomes. Zooming into how digital was integrated as part of the 6-minute walk test. I think this morning already Dimitri talked about the 6-minute walk test, which is, as you all are aware, a clinical validated endpoint in many neuromuscular indications. Knowing this, CMS is a disease where not all patients can walk, so we zoomed in here on the patients, the ambulatory patients. And in this 12-week study, we were able to demonstrate that those patients on the 6-minute walk test had a median increase of 50 meters. Now, you can wonder, what is 50 meters? Many of you are probably walking regularly. So if on a Sunday you walk 6 kilometers an hour, a 50-meter increase on 12 weeks is actually walking from 6 kilometers an hour to 6.5, which is almost like you need to run to catch your bus. So in that sense, this 50 meters is quite meaningful. We were not only able to demonstrate that there was an increase in the distance, because you can wonder, they can walk faster. It's like a sort of a forced setting in an in-clinic setting. But that's where the beauty came in of the digital health technology. Those patients also wore the Clario Opal device, so they were wearing sensors throughout the body, and we were able to measure things like gait, cadence, etc. And that's actually what helped us unlock a signal, because as I told you in the beginning, we sort of picked other scales from MG, like the QMG and the MGL. But what we noticed when we unblinded the data is that those two scales actually were confounded by placebo. We do not necessarily see that here at the 6-minute walk test, but the more powerful data actually came with the cadence data, because what you see in the cadence data is that we actually saw an improvement with the cadence. So like I said, patients can walk more during the forced in-clinic assessment, but it's the cadence that actually tells you not only that they walk longer, but also how, like in the sense that how smooth or how efficient they can walk. Cadence is unconscious behavior of the patients. So here you can also see that they did not only walk longer, but also that they actually were able to have more efficient walking. And that actually ties back to the mechanism of action of our molecule, which I talked about and which I started with in the beginning. So we were very excited to see those results, which then helped us reflect upon the next steps. And then I hand over to Jain to walk us through the in-clinic, to the remote.
Super, thank you, Kim. Yeah, so I'll now briefly go through some of the results that we saw from the real-world monitoring portion of the study. And so, as others have shared, getting patients to comply, especially in this situation where these are very heavily burdened, their symptom overall constellation is pretty heavy, but we were really pleased to see that with a combination of feedback to the sites and clear materials that we're sharing with the patients about why we want to do this and how it fits into the rest of the study, we saw that actually compliance was very, very high. So here we're just showing on the y-axis, each row is an individual patient, and across the horizontal axis, we're showing the compliance over time. And the darker green, the higher they are complying. And so we did get some feedback. As Kim mentioned, we also asked at the end of the study what that experience was like. Again, part of our aim here obviously is to be optimistic and think about what is the protocol going to look like in the phase 3. So we wanted to be aware of any potential issues or any potential hurdles that were holding patients back from complying as we would like them to. And so, yeah, for some of the patients, it was maybe not the most comfortable. Maybe it was hard to integrate into their overall life. But all of the patients understood why we were doing this, and all of them reported willingness to actually do it in the next phases, which is super important because many of these patients are actually rolling over into the next phases. So we moved forward with pretty high confidence that we could do this in future, and also that the data that we were seeing was going to be something that we could dig deeper into. And so when we took a first look at what the cadence looked like in the real world, and this is just cherry picking one of many, many parameters that were derived from the real world. So here we're looking at their preferred cadence. We also saw that the treatment group were improving their cadence over time in the study. And this is just cross-sectionally looking at the treatment and placebo groups over the 3 post-enrollment visits. And what we were also really pleased to see is that we also saw really good correlation between what we were finding in the clinic, and so this is the cadence that we saw in the 6-minute walk test, versus their preferred cadence in the real world. And obviously the preferred cadence is going to be slightly slower in the real world, as we've seen in many other talks. You know, nobody is doing submaximal tests in their everyday life, but nevertheless, that overall unconscious improvement in their gait quality was visible. And so we were obviously really, really pleased to see this and wanted to dig in further. And so coming back to our bigger evidence strategy, you know, we talked at the beginning about how the heterogeneity of CMS means that we really need to take a holistic view on all the different aspects and symptoms that these patients are experiencing. So we took a combined view of all the different kinds of outcomes. So this is the in-clinic functional mobility in dark blue, the whole constellation of measurements that we were trying out in red, plus the real-world measurements in pale blue. And we were really pleased to see that when we looked at the effect size versus significance, or a classic volcano plot, we could see that actually some of the digital measures were even outperforming those traditional clinical outcomes, in particular cadence, which is why we're sharing further data on that today. So great, you know, we felt that we could make a decision for CMS. What we're not talking about here is all the work that we did looking into individual patient trajectories. As a very, very small study, we're always going to be slightly wary of group effects. And so we did a lot of work looking at those individual trajectories to really decide whether or not we were seeing an overall improvement for these patients who are receiving treatment. But in the end, our decision was that, yes, this constellation of evidence was something that we could make a decision on to move the molecule forward into phase 3 in CMS. We are currently in discussion now with the FDA about what that phase 3 protocol might look like. And the evidence that we're presenting to the FDA is exactly what we've talked about today. You know, that the measures that we are collecting are meaningful, that there is a demonstrable impact on functional improvement in the clinic, and that follows through into stable improvement in the real world. And that overall holistic package is what we've strategized from the start. And we were just so pleased to see this move forward. And with that, I'll hand back to Kim to wrap up.
Thank you. And so, maybe tying back to Kara's presentation this morning, where she asked us to reflect upon the questions today, tomorrow, together. So with this presentation, we really wanted to confirm that this strategy that we presented to you, like the holistic evidence generation of doing the interviews, generating the data, making sure that we look at what is clinically meaningful for patients, getting the right measurements in place, integrating DHT. That actually is not only something that we do for CMS, but hopefully I inspired you that through our mechanism of action for 119, this was the right experiment for us to do. It did not only unlock this indication, but it actually unlocked the potential of our molecule to other indications where we're also applying what we shared with you today. And so with that, we do that through argenx style in co-creation with VivoSense, and we're very proud to share our joint learning with you today. So thank you very much.
Any questions? Yes.
Thank you for the nice presentation. I have one question about the demographics of your study. Do you have any details on the demographics, like the age, sex of the patient that you had, and did you see any difference there?
So this was a global study, and as far as I recall, it was like more of a diverse population. I don't know whether you can further speak to. There were no specific like male or female differences.
But yeah, as we say, it was an ultra rare disorder, so we were just pleased to enroll anybody that we could. So we tried not to place any restrictions in that regard. But it's certainly very hard to interpret, you know, those kinds of different when the groups are so small. But we certainly looked into it. Yeah.
All right. Thank you so much.
Thank you.
Thank you for that overview and for the progress update. And that's something that's super nice to see how the work sort of matures, and over the years we're able to follow and see how the impact is now being made on different drug development programs. And that is a nice